Hi everyone. So welcome back to a new seminar. So today's speaker is Lukas Stelzl. So Lukas is a group leader at the Institute of Molecular Biology in Mainz. Also uh, part of the soft matter group in the Faculty of Physics at the Johannes Gutenberg University in Mainz. So he has a broad interest, and today he is going to describe a bit of his current work and interest in molecular simulations, multi-scale, coarse-grained method development, and gene regulations. So, Lucas, so please, the floor is yours.
Thank you very much, Adolfo. And it's really nice to kind of be here today. And I think, my scientific career, like interacting with the Polish computational biophysics community, has been really, really instrumental. And I think very, very lucky to count many colleagues there among my friends. So today I want to talk to you about gene regulation and how condensates could possibly play a role in there. And this little movie is not directly about gene regulation. It's like a movie of a coarse-grained simulation with quite a bit of chemical detail of a protein forming a condensate. And this is sort of typically the kind of systems that we are looking at. With this, I want to, before we kind of come to the simulations, I want to give you a sort of a brief biological background. So many of us, or most of us, are aware that a central process in biology is copying DNA to RNA in gene expression. First step is transcription that ultimately leads to our mRNA being the template to constructing a new protein. The RNA polymerase II is a central molecule in there. It walks along the DNA and transcribes DNA into RNA. And already some years ago, there have been very interesting observations that RNA polymerase II seems to cluster in nuclei. And RNA polymerase II, the structure has been resolved already with structural biology techniques, and it has a folded domain which does most of the enzymatic steps. And it also has a repetitive sequence that if you kind of were to linearly arrange it, it would be enormously extended. So this disordered sequence has the potential to play important regulatory roles. In particular, people think that it could interact with other disordered protein domains in a process that is often referred to as liquid-liquid phase separation, and start to form phase-separated condensates. And this is a very attractive notion that would sort of explain many interesting observations in gene regulation, for instance, how an enhancer site that can be very far away from the start site of transcription can be brought physically close to the start site of a transcription process. It would provide a physical model of how promoter-enhancer interactions can work. This also goes back to older ideas, very precise ideas about so-called gene factories that have been put forward by researchers in the United Kingdom, Peter Cook in Oxford, for instance, and then further developed in Edinburgh. So this is, I think, a very interesting idea. However, it's not quite clear what the structure of the RNA polymerase II CTD would be and how it would interact with other molecules to form such a condensate. And these phase-separated condensates, I think it's important to point out, these are really distinct chemical environments. So there would be a dense condensate phase of proteins. There's, of course, still water in there. And that's a sort of outside, there's like a dilute phase of such proteins. But there's no membrane marking the boundary. This idea has been also taken further, that there's some evidence that you might not only have condensates that really start to assemble things for transcription regulation, but there could also be condensates that that could enable the actual copying process. So that would sort of be so-called gene body condensates, and they would, for instance, support the elongation and potentially also the processing of RNA. So here, really, the DNA would be copied and used as a template to RNA synthesis. So the overall idea is that in the initial step, maybe the RNA polymerase CTD, which can in principle, this is heavily post-translationally modified, particularly phosphorylated, that initially this would be unphosphorylated, then it would become maybe phosphorylated on serine 5 positions in the disordered CTD domain. And then in this gene body condensates, there would also be perhaps serine 2 phosphorylation. So these phosphorylation marks have been studied intensively over time. So these are well understood things, whereas the condensates are a newer idea. So this, I think it's a very cool idea, and it's, I think, a very attractive hypothesis to consider that there are really two condensates, that one is helping to assemble everything for transcription, and then the second one really enables the transcript being elongated. But it's not at all clear, I would say, whether these promoter and gene body condensates really do coexist. And if so, how would they actually stay demixed? Because the components in here actually quite similar. So why won't they just kind of become one big condensate? So this is, I think, is a big question. I think this has not. I think this really requires a more work from a physical chemistry perspective. So in this talk, I will address three kinds of questions. One, to try to understand a bit more better how RNA polymerase II CTD interacts with condensates and other proteins. This in the condensation or phase separation field, this is sometimes referred to as the molecular grammar, to try to understand which kind of local interactions can be important for phase separation. And then we try to understand how promoter and gene body condensates can coexist and how they stay demixed. And lastly, I will briefly also investigate to what extent these different condensates can actually recruit different molecules or they have different physiochemical properties. Because if they really are supposed to provide for differential regulation, you would expect that they have some differences in how they recruit molecules. So in my group, we typically try to run simulations at multiple different scales, starting with atomistic simulations of single chains, which are still computationally tractable, simulations of protein complexes, also, I guess, it's quite feasible. And then pushing things a little, we can even simulate whole protein condensates at all atom detail. And if time permits, I will show you an example of this. This is, of course, not so easy, but it has it's potentially very interesting because you can really see the effects of small chemical changes in the context of a big condensate. So I really understand how a specific side chain might interact with another molecule to affect its recruitment, or how a post-translational modification might modify such a condensate. And in this picture, I have shown one of the chains in sort of glorious atomistic detail, and also some of the water molecules in the back are shown. So I could not include all the detail here, but just to illustrate that really we can, in principle, follow the movement of all the water molecules in such a system. But of course, at some point, the computational cost of such a simulation becomes, of simulating such a system becomes prohibitive. And then we need to simplify the system. typically what's done is coarse graining. So one groups some particles together to new particles, and this then enables us to access the long time and length scales needed to really see molecules phase separating spontaneously in our simulations. And I think it's important to point out that this simplification is not just computationally advantageous, but I think this always provides also an opportunity to learn something deeper about what are the important factors driving the system, like what can we neglect in our description, and what do we really have to include in our description to simulate what we care about. And ultimately, we can simplify these descriptions further, perhaps building Markov models, or like even more simple particle-based, sort of like losing all particle-based description, or perhaps using even simpler particle-based descriptions. Many particles, many atoms or residues correspond to one particle, and that with that one can really ultimately reach cellular time and length scales. And most of the work I'll show today is the PhD work of Aya Changiratsi-Visadan in my group. And to start, we wanted to understand a little bit more about the CTD, the disordered domain of RNA polymerase II. So our collaborator, Markus Zweckstetter in Göttingen, he measured some very exciting NMR data on RNA polymerase II. And I really think the exact details here do not matter so much for this talk. But I think what's crucial is that he found contacts between different amino acids, NOE contacts, some well-defined contacts, for instance, between prolines and tyrosines. And this is unusual for a completely disordered protein. So that begs the question whether the RNA polymerase CTT is actually completely disordered. In addition, he was also able to measure global structural parameters such as the hydrodynamic radius of RNA polymerase CTT. So what we wanted to understand is to what extent there could be some ordering in RNA polymerase II CTD, and to really be sure that a description as a disordered protein is a good way of making as kind of, it's a good way to think about this really important molecule. And we were able to generate atomistic models. There's some ensemble of this shown, and we were able, using a Bayesian framework that I developed while I was co-developed while I was a postdoc in Gerhard Hummer's group. We were able to refine this very slightly, like these tiny changes in the weights of the individual structures, and then we can account for most of these NOE distances. So these experimental contacts can be translated into a distance cutoff, saying that this particular pair of protons is within a cutoff of about 5.5 or something like this angstrom. And most of these contacts seen in the experiments are actually can be accounted for via very slight refinement of the weights of individual structures. So that would suggest that the initial atomistic models were maybe somewhat too loosely packed, but they were already pretty good in capturing some of these contacts. And at the same time, we also included global characteristics, such as the hydrodynamic radius in our refinement procedure. And overall, we then arrive at integrative models of this largely disordered protein, but then that still has some local structure. And when we then look at the structure, there's some interesting things to be learned. And this is also maybe already directly visible in these this NMR spectra where the Stelzl lab identified these tyrosine proline contacts, and these tyrosine proline contacts are also apparent in our structures, as well as if you were to use these NOEs for more traditional structural determination protocol, where you really try to find one single structure accounting for all of the data, or if you take a simulation-based approach where you try to build models and then slightly refine the weights of the models to better account for the data. But both things highlight these tyrosine proline contacts. And what's very interesting about this could also play a role not just to drive the phase separation of an RNA polymerase II CTD, but also for its interaction with other molecules, but in its biological function. So this is a cryo-EM structure of the mediator complex in RNA polymerase II. And here it is the DNA, and there's actually little fragments of the CTD are resolved. And there you actually see these tyrosine proline contacts. And this the paper by the Stelzl lab has just been published, including our contribution to this. And with this kind of motivates us to go further. I mean, we now have some atomistic insight into what's going on in the interactions of RNA polymerase II CTD, and we now use a very simple coarse-grained model where each residue is a single bead, and different amino acids have different interaction propensities. And there's also an implicit solvent model and using the Hückel electrostatics to also describe the interactions or the effect of charges in the system. So some amino acids are, of course, charged. And then when we look at the contacts in a condensate that forms in such a simulation, we see, for instance, tyrosine contacts are very prominent. That's not surprising that often these relatively large aromatic residues are well known to important for phase behavior. But we also see tyrosine proline interactions. And I think here really the insight from the atomistic modeling and the experiments is really crucial, because I think we otherwise would not have been on the lookout for these interactions. But I think it's very nice to see that a much simpler coarse-grained approach actually captures some of these trends. And then just a brief note on the simulation setup. We of course sometimes run simulations in big boxes with really droplets, or other times we kind of simplify the system somewhat, kind of to simulate things in slab geometries to better enable the equilibration across interfaces, and also to have less of curvature effects. And another thing I want to point out is that if we compare our simulations, coarse-grained simulations, with many atomistic reference simulations of fragments, we actually see that in atomistic simulations, these fragments are also have attractive interactions, as they do have in the coarse-grained simulation. And some of these tyrosine proline interactions and tyrosine interactions are also seen in these atomistic simulations of small fragments. So overall, I think we have a reasonable simulation model to proceed. And another thing that was identified by the Stelzl lab was that this RNA polymerase II in vitro phase separates more strongly at higher temperatures than at lower temperatures. And this is something that can also be captured with a variant of this coarse-grained simulation model, where previously a temperature-dependent term was parameterized. We just took the previous parameterization, and indeed we find for RNA polymerase II that at lower temperatures at some point you no longer have a condensate, whereas at higher temperatures we actually have a condensate. And what's very exciting about this our colleague of ours, Jan Padeken at the Institute of Molecular Biology in Mainz, he's actually interested in similar kind of phenomena, and he's studying them in C. elegans experimentally, and he can really do experiments with C. elegans with these kind of really tiny worms, which are like a very important model system for biology. And when he looks at C. elegans embryos in these worms at different temperatures, he finds that the condensates, or the foci that we interpret to be the condensates, these are RNA polymerase II tagged with GFP, become more prominent at larger temperatures than at lower temperatures. So that would suggest that indeed also in vivo RNA polymerase II CTT show, or RNA polymerase II shows a lower critical solution temperature. And that's actually something that's also captured in the coarse-grain simulation. So I think that's, very encouraging, this agreement between experiments and simulation. I think what's also very interesting and exciting about this is that other condensates in the nucleus do show different temperature dependent. They typically dissolve at higher temperatures. So that would suggest that there really are multiple distinct condensates in the nucleus. In my introduction, I already mentioned that phosphorylation is really important in the regulation and the transition of transcription initiation to elongation. So we also run simulations now with this coarse-grain model that we have compared to reference data from experiments and more detailed simulations, and also to the temperature dependent in C. elegans. And we now phosphorylate some chains, as would happen in reality. And what we see, if we partially phosphorylate the chains, they typically still like to go into the dense phase. Now, as you phosphorylate the CTT chains more, and sort of the degree of red of these chains indicates how much they are phosphorylated at serine positions in these heptad repeats, and we always here phosphorylate the fifth position in this heptad repeat. When more of this fifth position get phosphorylated, then more of these phosphorylated chains go into the dilute phase, which is in line with experiments. And then if we fully phosphorylate these chains, then there's no longer a condensate. Again, this is in line with experiments. And we see that there's like a steep transition with the degree of phosphorylation and how many of these chains are in the dilute phase. So this steep transition would also indicate that this could be useful for biological regulation, where you really want to maybe have a strong response to phosphorylation to really delineate different aspects of this transcription process. And just very briefly, this is data from the Zweckstepperlab that kind of a very important paper in already a couple of years ago. They showed that the RNA polymerase II can indeed form condensates, and then when a kinase acts on these condensates together with ATP, then over time these condensates dissolve, and that's we also see that if we really phosphorylate all the chains, then the condensates dissolve. Another paper around the same time also showed a very interesting thing, that if the RNA polymerase II CTT together with elongation factor called HRD also can phase separate. So this HRD is part of PTFP, cyclin T1, it's an important part of transcription elongation. And together with PCTD, it forms condensates, and CTD together with HRD do not interact so much. We find that if we just have CTD and HRD, there are some interactions it's not very strong. If we then phosphorylate the CTD to be PCTD, then we really see very strong interactions. If we look at how the residues are interacting, we see a lot of interactions between positively charged residues in HRD with phosphorylated serine. So lysine phosphoserine interactions, arginine phosphoserine interactions, which of course makes sense. We can then quantify this, and Aya was able to determine phase diagrams for this, where she varied the she started with a fixed amount of 100 HRD chains, and she added PCTD chains. And the more chains she adds, the bigger the condensate becomes, but at some point all the HRD chains are happily interacting with PCTD, and then additional chains just go into the dilute phase. So there are negligible homotypic interactions in the system, and electrostatics really drive cross interaction and self-repulsion. This can also be better understood by comparing to the mean field theory. This was very nicely done by Jasper Michels at the Max Planck Institute of Polymers in Mainz, and he could show or can fit his mean field model to our simulation data. And again, the slope of the tie lines also highlights this is an associative phase separation. So this makes sense, and this is also in alignment with biological data on the interactions of PCTD and HRD. We can now sort of take this a step further and really bring all three components together. So CTD, which is mainly important, it's at least the picture for transcription initiation, HRD, which is part of an elongation factor, is a disordered part of an elongation factor, and PCTD, which also is important for the transition from transcription initiation to elongation. So when PCTD is partially phosphorylated, everything can nicely mix together. We get one dense phase, and if Aya increases the degree of phosphorylation, then there may be some substructuring in the dense phase where PCTD and HRD are more towards the interfaces, and when she increases the phosphorylation further, we really start to have kind of distinct environments or distinct sort of regions where CTD is more prominent, and other regions where PCTD and HRD seem to cluster, and further phosphorylation really leads to the emergence of really distinct phases. So one phase for PCTD and HRD, sort of the blue and the red beads, and another gray phase that's for unphosphorylated CTD. So the red and the blue phase would be for transcription elongation, and the gray phase would be for transcription initiation. And you can also see this very nicely in these density profiles, or this sharp transition that is actually a sharp transition with the degree of phosphorylation can also be nicely seen by when Aya computes the size of the biggest CTD cluster. And again, this idea that there's a sharp response to phosphorylation could also be important, or we feel that could be very important for the actual biological process, so that really after a certain phosphorylation threshold is reached, then you really have potentially two different phases important for initiation and elongation. So we wanted to check that this is really this is consistent. So we Aya determined looked at the different ways of how two liquids can arrange themselves depending on the interfacial tensions. So of course you could have one liquid surrounding the other liquid. This depends on which one has the strong interfacial tension. The one with the higher interfacial tension would be at the center of such a droplet. Of course, two liquids can also be totally apart from each other, sort of not interacting at all, entirely separate. And that's kind of in a way the picture that I have drawn in the introduction, where these condensates would be entirely separate. What's also possible is that there could be some partial engulfment or some contact angle between these two liquids. And the relative interfacial tension of these two liquids, and also for the interface between those two droplets, would determine which of those regions is the more favorable. So in this region, you get the gray in the inside. In this region, you would get the red in the inside. Here, in this region, you would get these two droplets in contact with a certain angle, and here the angle can be computed. And in this region, these condensates would be entirely separate. So I was able, using method developed originally in the polymer field, looking at the fluctuations of the interfaces to determine these interfacial tensions for simulations of CTD, also for here this the interface for between PCTD, HRD, and the surrounding solvent, and also crucially the interfacial tensions between the two dense phases. And here she built on some very nice work Friederike Schmidt was involved with, and Friederike was really instrumental in getting this to work. And one caveat or one concern could be that this is maybe only true for slabs. I also verified that this also works for droplets, and we get similar values of the ratio of the interfacial tensions. And all of these would suggest that indeed there could be a gray phase, important for transcription initiation, that would be surrounded by this mixture of blue and red. Maybe another thing that could be interesting here to note that both the slab and the droplet simulations would suggest that our setup is not so far away from a partial engulfment morphologies, and perhaps by changing the composition of the droplet slightly, you would get partial engulfment. We can also see partial engulfment if we just have not enough PCTD and HRD chains around, but we can also see it by adding more components. So this is sort of then a simulation of a big droplet and a cut through this to show you that really we have this core of CTD and it's surrounded by PCTD and HRD. So in simulations, you can see both this engulfment and this partial engulfment morphologies. And this, especially the engulfment, is entirely consistent with our interfacial tension computation. The partial engulfment, there would need to be some likely some small changes in the composition of the two droplets, or if you don't have enough material, then you also get partial engulfment. And what's super exciting about this is that Jan was able to, Jan Paddington at IMB, was actually able to look for these things again in C. elegans embryos. So looking at embryos in these worms and looking at a stage of embryonal development where transcription has already started. And then he had antibodies against phosphorylated RNA polymerase II, CTD. And maybe let me draw your attention to this column here in the center, where he specifically looks for serine 5 phosphorylated RNA polymerase II. And sometimes you really see these kind of rings. So those would potentially correspond to these kind of morphologies where you really have a ring of RNA polymerase II surrounding phosphorylated RNA polymerase II surrounding unphosphorylated CTD condensates. Sometimes you see something that looks more like partial engulfment. Again, said this is also consistent with our modeling. And he also looked at serine 2 phosphorylation. This would be further along in the transcription elongation process. And what's also interesting here about this, again, this shows similar profiles, and typically serine 2 phosphorylation and serine 5 phosphorylation are kind of connected. So this could indicate some more sort of more organization of the transcription process. But I think the take-home message here, what's really most exciting is that really one sees these kind of ring-like structures or partial ring-like structures. And this is also something that our simulations have suggested. And I think without the, I think it's super exciting to see that this coarse-grained model predicts some shapes that then can really be seen in an organism. So this would suggest that indeed there can be different phases or different condensates for different part of transcriptions, or for initiation and also for elongation. These could be sort of organized in a sort of engulfment or partial engulfment. But if these really are distinct phases, we would also expect them to have some differences in properties. And so I looked at how the conformation of PCDD changes in these different phases. And just very briefly, the squared radius of gyration increases when PCDD is in the unphosphorylated phase. So it sort of elongates, probably also by partial self-repulsion because it's negatively charged. And it tries to elongate. So one looks at the radius of gyration tensor, the perpendicular component to the interface is increased. So potentially it tries to extend maybe into the phase it likes to be. So it orientates perpendicular to interface to maximize its interactions with the PCDD and HRD phase. So I think what's interesting, I guess what's the most interesting biological aspect of this is that this really shows that these interfaces, these condensate phases really modulate the structure of the molecules, the global structure of the molecules in these phases, and this can then further down the line give rise to differential interactions of these phases with other molecules. And we have also tried to see this in our simulation. And if we add FUS, which is known to interact, can act as a transcription factor and is known to interact with promoter condensates and interacts less with phosphorylated CTD than unphosphorylated CTD, we really see that the density of FUS aligns well with the density of CTD, and where there's a lot of PCTD and HRD, the FUS is relatively depleted. So it can preferentially interact with CTD also in the context of a multiphasic condensate. So this really suggests that these simulations and this overall model of how transcription could be regulated can also account for some of the differential recruitments of other factors to these different condensate phases. So with this, I want to kind of wrap up the main part of the talk, and if there's time, show you like 2 little vignettes of other things going on in the lab. So what I've shown you is that phosphorylation can act as a switch, which gives rise to different dense phases once the threshold is crossed. The CTD is largely disordered, but there's some residual structure that we can understand. Some of it may be important to bind other factors, such as the mediator complex in transcription initiation using this tyrosine proline context. We can compute interfacial tensions from these microscopic simulations to better understand these morphologies. and transcription may indeed occur in condensates, which have these sort of core shell or partial core shell like structures. And this is also borne out by experiments by my collaborator Jan Padek, and based on our simulations, which is super exciting to us. So very briefly, especially since this is the seminar hosted by Adolfo Pomo, I also want to show you a slide on how we can model both folded and structured domains in condensates. And then if time permits, just show you that we can really resolve on the atomic scale important contacts of how a condensate can recruit a peptide. So just very briefly, this is work by Xiaofei Ping in my group. This is a condensate of the protein TDP43, which is very important in neurodegenerative diseases. And she has been simulating this both with elastic network models for the 3 folded domains in the condensate and also with Go-type models. And these are quite challenging simulations. What's very encouraging about this, that she really sees chains going in and out so here she plotted the density over some segment of the simulation, sort of a chymograph type plot. And she's really, you can see some chains going in and out of the condensate, and she can then start to generate density profiles and then phase diagrams from this. And she is also doing this with Go-type models to also better understand how flexibility of the folded domains could be important to in the formation of these condensates. And the last thing, so this is just a little movie, and you really see these cyan disordered domains interacting, and then you have these globular domains in orange and magenta and green. And the very last thing I want to show you is that we can really use these multi-scale simulations also with a lot of chemical detail to better understand how peptides or other proteins might interact with a condensate without going into the biology here of the process. This is a condensate that's important in small RNA processing in C. elegans. This is joint work with a collaboration with the lab of René Ketting. In Martini simulations, a part of RDA2, which is important for getting the right enzymatic activity into these condensates, or foci in vivo, interacts with these big droplets. Gaurov, which is student working on this, identified interactions between tyrosines in the peptide with arginines and lysines, and this seemed to be some sort of important sticker interactions that has been very prominent in this in the field of phase separation, also based on important work in polymer science, that these could help to recruit this peptide to this condensate. And he cut out a small part of the system, backmapped this to all atoms. This is about 800,000 or 900,000 atoms, particles. And here, this is the blue is the condensate, MAT16, red is peptides. He has been able to run this for microseconds, both on local resources and then further simulations on folding at home, which is a very large resource where people donate their computers to for scientific calculations. And then, for instance, he can follow contacts between these arginines and lysines and tyrosines in the peptide. For instance, like zooming in on the arginine-tyrosine interactions. Sometimes the arginine is quite far away, and then over time gets closer to the tyrosine and forms a sticky contact. Sometimes it's also the other way, that over time the contact breaks. So he finds a lot of cation pi arginine interactions, more so than for lysine. And maybe one effect is that the arginine also has this additional hydrogen bonding potential for the side chain. And so this could account for some of the difference between arginine and lysine. And what's very cool about this, the lab of Sebastian Falk in Vienna has tested this in vitro with mutations, and indeed arginine to lysine mutations reduce RD2 binding, and arginine to alanine interactions lead to a loss of RD2 binding. So that was all I wanted to say for today. Thank you very much for your attention, and it's really cool being here. I particularly want to thank Aya for the great work, and she's really in the lead of the phase separation RNA polymerase II project. Kuma Gaurav is working on the specific recruitment in condensates with all atom resolution. Xiaofei Ping works on TDP-43. Also want to thank Emanuele, Mahesh, Lucia, Yehor, Max, Vasilis, Dennis, and Cyril. So really wonderful colleagues to work with, and very excited to continue to work with them. My collaborators, Jan Padikem, sort of microscopy experiments, Jasper Michels for the mean field theory, Markus Zweckstetter on the NMR experiments, Lisa Pietrek for simulations method development, particularly for the atomistic modeling of the disordered chains, Sonja Hansen for the collaboration on holding at home, and Friederike Schmidt for the collaboration on the more the polymer science aspects of this project, and also for hosting our group in the Comet supergroup. And thank you very much for your attention, and I look forward to your questions.
Thank you very much, Lukas. So now I'm quite sure that there will be many questions in the audience, so please raise your hand. Yeah, it seems I raised my hand. Okay, please, Jens, go ahead. I think you can.
Yeah, hello from Dresden. I'm Jens Sommer from Leibniz Institute of Polymer Research. Thank you very much for the nice talk. Great to see how far you can get with this systematic cross-graining in the proteins. I have a question. Maybe you said this, but I missed somehow this aspect. What determines the position in the DNA where the enhancer is formed? I mean, the gene body condensate apparently, or at least to my opinion, will have some affinity with the RNA production.
Yes.
But the enhancer position, what determines this one?
So my point of what I would think is important is that the enhancer and the promoter, right? You have specific DNA sequences that are key, that are important for binding transcription initiation, transcription factors and things like this. And there are, of course, also other effects such as looping of DNA segments, where you have these CTCF proteins, which kind of forms these extrusions. So, of course, there are higher order effects in chromatin structure will there be super important, also chromatin modifications. And this is, of course, something that we are not touching at this stage yet. And this, of course, there's some really exciting simulation work going on with this, where this is also explicitly taken into account. So I would say that we're kind of trying to work from one way up, and I think other people are working from down. And I think soon we'll meet and get really comprehensive multi-scale models of what could be going on. And I think there's lots of exciting stuff there, in my view. For instance, I suspect, and I saw a preprint this morning that goes into this direction, that, of course, these promoter enhancer contacts, they will also be sort of dynamic and stochastic, and they will not always be in contact. And I think another very interesting aspect of this is, of course, that the binding sites on the DNA, I think, right, this is not just a very small segment, but there are multiple binding sites next to each other, where I think these factors can cluster as well. There's also very interesting experimental work from sort of more in vitro work suggesting this from Sina Wittmann and others. So yeah, I think our models are not so complete about this yet, but I think there's a lot of cool stuff to be done there.
Yeah, because I think that's a very, very interesting question in general.
Yes.
And I think I read also about a hypothesis that the enhancer droplets, these condensates, can help in the target search of the transcription factors. So they are positioned outside of the coding regions and collecting transcription factors, which can be then exposed at a certain signal, and they are close to the genes which should be activated. Can you comment on this? Or
I mean, it sounds very cool, but I think, right, our model doesn't even include DNA yet. So I think, but I think this is sort of one of these motivations, I think, why we really want to do this kind of things to get there eventually. But I think I really also think that going the other way is equally important, right? So kind of have these even more coarse-grained models where people start to add bit more what's known from the molecular side. So I think that could be very cool.
Yeah. Thank you.
Thank you.
Okay, let's continue with the discussion. So more questions. Anybody can raise the hand. If not is the case, so I will ask one question regarding this coarse-grained model. So in the coarse-grained model, basically you include, so the dominant interaction is going to be electrostatics, right? So that's one of the components. And the and regarding so these are implicit solvent models, yeah. So you have the bihookel interaction, so that accounts for that. So what about the flexibility of those chains in the coarse-grained model? Is it parameterized, or it's the same for all the chains?
So it's so the beads, right, have some tendency of how much they like to interact with each other. There's a very nice study by Arash Bashman where he showed that the stiffness of this model varies locally. So there are some differences. So that I think what he concluded that, for instance, a uniform, like a persistence length for the whole chain is maybe not such a great idea, and that you really would have to look at this segment-wise. But yeah, so I'm not sure whether kind of really chain flexibility is taken into account.
Okay, but
best possible way in this model.
But when you construct these residual contacts into the condensate, so you introduce some kind of flexibility in the full condensate, right? So that's how somehow the model reads the information from the NMR. How this?
Sorry. So we did the NMR. So microstructure did the NMR. We ran these atomistic simulations. We kind of did an ensemble refinement against the NMR data to sort of, in a way, I mean, without any ensemble refinements, we could already kind of capture, I think something like, I don't know. I don't know the exact numbers, but kind of more than 2/3 or 70% of all the NOE distances without any refinement. With the refinement, we can capture them slightly better. I think only 2 of them we can not fully account for in our model. So that, in a way, this gives us then an idea of what contacts are important. And the cool thing is that the coarse-grained model that has been parameterized by the group of Chita and Mittal, it does actually already kind of produce these contacts. So we did not tweak the coarse-grained model. But of course, in an ideal world, right, maybe one would really kind of maybe take the information from the atomistic simulation into account by kind of fine-tuning the coarse-grained model. Here we kind of used a different higher resolution pieces of information to get a better feeling that the coarse-grained model is a reasonable coarse-grained model.
Okay. No, I was just trying to imagine if for the cases of supracortical model of chromatin, somehow they introduce this kind of contexts which are between different parts of the complex system. So maybe it's a good way also to upscale the system, perhaps.
Yeah.
Okay, so more questions. I think it was quite interesting and clear. So there are no more questions. But yeah, you have greetings also there. And so it's over 1 o'clock, and I think we will stop now. And we say you thank you, Lukas, and certainly it was a very interesting talk, and we hope to stay in touch regarding the simulations and other topics. And yeah, so
thank you so much.
Take care. See you guys. See you in 2 weeks. Bye-bye.